Greetings, ecologists. This is video 6. We're picking up where we left off in trying to answer the question, why are there so many animals of varying body size? So at the end of Friday's lecture, we talked about the heterogeneity in grass quantity and quality in savannas. And today, we're going to talk more about how that helps explain this pattern. So I shared with you as a reminder that these are images of all the different large herbivores. I'm not including small mammals, nor am I including insect herbivores, but what I kind of consider mega herbivores, though that's a subjective definition, from hare the whole way up to elephant. And you'll notice that there is a pattern here. We don't have really two animals that are identical in body size. There are gaps, there are partitions of body size. So we're going to answer these two questions. We're first going to talk about why there's so many animals as a reminder, and secondly, why do these animals vary in their body size? Evolution created this pattern, and we want to know what selective forces might have led to these differences. And it will take us, in fact, the whole course to keep teasing apart this question, but I want to give you a little bit more information today. So we firstly know why there are so many animals in general, because savannas and grasslands produce so much fairly digestible food. But today we want to explore the second part to that question. Now remember, this is what Kansas used to look like only 14,000 years ago. During the Pleistocene, we had all kinds of creatures, and they indeed varied in their body size. So to give you an idea of scale, the native equids that we had, horses, woolly rhinos, this is a ground sloth. To give you an idea of scale, here's a human scaled to a ground sloth. So you realize these were mega herbivores. This community also had a lot of complex interactions, some of which will relate to predators and prey we'll talk about later, but we only have one remaining antelope species left in North America. This is the American pronghorn antelope. All the rest are deer species, not antelope, which is quite the opposite in Africa. There, by the way, there only one or two deer species, and everything else is antelope. But at any rate, this is the fastest land animal on the North American continent. You might wonder why. Well, that's because it used to be chased by a Native American cheetah. This was a species that went extinct about 12,000 years ago. So let's remind ourselves a little bit about this forage that is available to these large herbivores in the savanna, and we need to remind ourselves what cellulose is. So I mentioned to you in the last lecture that cellulose determines the quality of the forage, and I want to explain a little bit more why. So the first thing we need to remember is that cellulose is a long-chained carbohydrate made of glucose, and those glucose molecules are really, really tightly held together by hydrogen bonds. You might remember from chemistry explorations that hydrogen bonds are opposite attractions between positive and negative charges. So in a cellulose molecule, this is a glucose molecule. Count them carbons, 1, 2, 3, 4, 5, 6, C6, H12O6, that's sugar, glucose. These are bonded together by hydrogen bonds between one layer of glucose molecules and another. So the hydrogens have a slight positive attraction, and the hydroxyl groups have a slight negative attraction, and they due to hydrogen bonding, come together. So because these molecules can get so close to one another, these hydrogen bonds get really, really, really strong. And therefore, it's very, very hard to cleave apart these glucose molecules. In other words, it's hard to digest cellulose. Along came, however, the evolution of rumination. So by watching the other two short videos about rumination, you've learned a little bit about that process. So as a reminder, rumination is when a cow provides essentially free room and free board for bacteria that help break apart the cellulose. There are certain species of bacteria that have an enzyme called cellulase. Remember, we name enzymes usually regarding what product they cleave apart. So cellulase is an enzyme that helps organisms digest cellulose. And the cow provides plenty of water, plenty of food in the form of cellulose, and a nice, warm, cozy environment. And it gets, in exchange for that, bacterial waste products. We call them volatile fatty acids. These are 3 of the volatile fatty acids that a bacteria produces as it breaks apart glucose in the form of cellulose. And you notice that there are a tremendous amount of carbon-hydrogen bonds left. In other words, a tremendous amount of energy left that the cow can absorb and break down. A cow does have the enzymes necessary to break down acetic acid and the rest of the volatile fatty acids. Now, the other interesting thing about rumination that you maybe were introduced to in the other videos and we'll discuss today is that bacteria eventually die and pass out of the rumen and into the cow's gastric stomach, and therefore provide a protein source in the form of their own bodies. So don't forget, bacteria are people too. They have proteins in their tissues that other animals can digest. So bacteria provide these 2 food resources: the waste products of fermentation and their own bodies in the form of protein. Now, bacteria take time to break apart and ferment cellulose. It doesn't happen instantly. So this cellulose has to be fermented by the bacteria in an animal's gut, and it's an ecological process. Therefore, the bacteria have to multiply and grow, they have to have the right conditions, and it takes a while for them to break apart and digest the material. So what I want you to realize is, if we think of this axis as time for an animal to eat something, to digest something, and this is the fermentation rate, you'll notice that cellulose takes a long time, up to 20 hours, to kind of fully ferment, where what we call a soluble carbohydrate, something that essentially dissolves in water, this would be sugar water, for example, glucose like in your drinks, or fructose, these things get fermented really, really rapidly. So this means that the amount of time the bacteria need to break apart cellulose versus an apple, or something with a lot of soluble carbohydrates, pectin is another example found in fruits, we have to think about what we call retention time. That is, how long does the food stay in the animal's fermenting chamber? Okay, so cellulose means we need long retention times. If we're breaking down something that's highly digestible, we don't need long retention times. So let's remind ourselves, as you saw in the previous video, what a rumen looks like. So here's our friendly cow with the esophagus just like you have. And then a cow doesn't actually have 4 stomachs. It has 4 compartments to a stomach, essentially. We have the reticulum, the omasum, the abomasum, and the rumen. All right. The first thing to note is that the material comes down the esophagus. Here's an enlarged version. Comes down the esophagus, passes into the rumen, and this is where most of the fermenting bacteria live out their lives, essentially, inside a cow's gut, essentially. And so this process takes time, depending on the coarseness or the amount of cellulose in the material. The higher the cellulose, the longer it takes to ferment. Eventually, this food material will pass through the reticulum. The reticulum is a series of folds that actually start to filter out the large chunks of material. So anything that isn't well fermented gets retained in the rumen, and it gets increased its exposure to fermentation. It stays there for a longer amount of time, and fermentation is allowed to continue. The particles that are highly fermented and are very, very small pass through the reticulum, down through the reticulum, and out into the omasum. The omasum, we're not exactly sure all of its functional roles. There are some materials that are absorbed through both the rumen wall, particularly the volatile fatty acids are absorbed through the rumen wall, but we think that there might be some nutrients also absorbed through the omasum. The key part I want to emphasize is the abomasum. That's the cow's true stomach. That's where hydrochloric acid is released, just like in your true stomach, to break down protein. So the material then goes into the abomasum and then out into the small intestine, etc., into the large intestine, and then out the animal's rear end. So the point is this: the cow's rumination process slows down the passage of food and increases retention time. It allows the material to stay in the cow for a longer period of time. That's benefiting the bacteria. It allows the bacteria to do more and more fermentation. Another thing a cow is really good at is rumination, which is essentially regurgitating food back up through the esophagus, and the cow's able to re-chew it again, break the particles down into smaller sizes so that the surface area of the particles increases and the bacteria have an easier time of fermenting, and then it swallows it again. So this circle back and forth is what we call rumination. And again, the reticulum is what essentially is a mechanism to allow the fermentation to increase for long periods of time until the particles are well digested. Some of the bacteria gets flushed out of the rumen and goes into the abomasum, and the cow then eats the bacteria. So while the cow provides free room and board for its renters, it also eats its renters eventually. Which is an interesting phenomenon. So you might have some questions after watching those videos. The videos I highlighted for you about rumination only are the animals that are in between these boxes. These animals are actually not ruminants. They are what we call hindgut fermenters. They do not have the ability to regurgitate their food, and they do not have a reticulum, omasum, and abomasum. They have a true gastric stomach, and they have a huge, huge hindgut. And they retain their food in a different format, which I'll show you in a few slides later. But all these other animals are like cows. They're ruminants. They can chew their cud. A giraffe can chew its cud. And so it's a key part about their digestive physiology that we need to understand. So let's contrast the hindgut fermenters, like a horse or a rhino or an elephant. Let's contrast them with foregut fermenters. So a hindgut fermenter only has one single chambered stomach, and it's also very adapted to eating cellulose, but it's because it has this massive long hindgut tube. That's where the fermentation occurs largely for a hindgut fermenter. That's where the retention time is extended. The fermentation occurs after the true gastric stomach. And so if we look at a photograph, you can see in a horse this large tube that it has. Essentially, a cow is a huge vat and a horse is a long tube, and they both are designed to increase retention time so that bacteria can digest cellulose. These are co-evolutionary patterns that digestive strategies that these animals co-evolved with to break down all this cellulose forage. Okay, so we can compare some of these large hindgut fermenters. This is a scale diagram to show the size of their gastric stomach compared to the size of their hindgut. And essentially, these are massive, massive long tubes. This part right here has been highly reduced in humans. That's called the appendix. Your appendix is now a tiny little pouch. Essentially, the food moves through the small intestine, it comes around the corner, and some of it gets pushed up into the cecum here, and this is an dead end, so to speak. So that really increases retention time. A lot of fermentation happens there. Then some of that material gets pushed out into the larger intestine and eventually out the anus. So in a capybara, that we saw this in a slide earlier, it's the biggest South American rodent. It's also a hindgut fermenter, eating grass and material. Look at its massive appendix. You and I don't eat as much cellulose as our ancestors, as our hominid relatives ate, like chimpanzees. So our hindgut fermentation is very minimal. In fact, sometimes our appendix gets blocked and we get an infection in there, and we can burst our appendix and send all that bacteria into our body cavity, which can be deadly. Okay, so we have a bit of an understanding now of hindgut fermentation and foregut fermentation, or the ruminants, and we now know that retention time is really, really important for an animal to make use of the cellulose and digest it. It can't digest it alone in its gastric stomach without the benefit of bacteria. So let's return to the pattern of body size. Body size really seems to be an interesting pattern all across the globe, and particularly in grasslands. So why do the number of species in an ecosystem follow kind of this distribution? So birds have different body sizes, mammals have different body sizes. This is the proportion of species with different body size. Notice there are lots of different body sizes across taxonomic groups. In other words, we should conclude immediately that evolution doesn't think that one body size is the best. It uses multiple body sizes as effective designs or effective plans for how to make a living, for how to survive, and to get your genes into the next generation. Well, let's contrast now body sizes of large megaherbivores in the savanna, and we're going to introduce you to something called the Jarman-Bell principle. So small-bodied animals, it has been observed in the field, really prefer high-quality food. They need just the live leaves or the tiny little berries on the plants. That is where they really feel happy. So this little Thompson's gazelle is grazing on this really short grass that is highly nutritious. Remember, it's highly nutritious because it's green and it's mostly leaf and has very little cellulose. Now, a body size like that also needs less total food, right? Because it has less tissue to support. So it needs high quality, and it focuses on high quality and less quality. So it likes leaves, short grass, rare things, and due to the fact that heterogeneity is scale dependent, it can perceive these resources at very small scales. So sometimes it can forage on individual leaves off of a plant rather than eating the whole plant. Contrast that with one of our favorite megaherbivores, the African elephant. It's extremely large body size, and it can tolerate very, very low quality food. In fact, it can eat trees. It will push over a tree, break off a branch, and it will be able to digest partially some of that woody material. But since its body is so massive, it needs a huge quantity. So the only way it can find a huge quantity is by eating some pretty cruddy quality, such as dead old stemmy grass and dead and dying trees sometimes. So the fact that its body size is really big means it's also constrained to eat some fairly low quality material. Okay, Jarman and his colleague Bell, they noticed this principle by just observing animals in the field. Now, let's try to understand why evolution selected for that pattern by linking this pattern to some of the digestive traits that we learned previously. So here's some data by Owen Smith and Cumming from 1993 that corroborates the Jarman-Bell principle. It's the percent of non-stem in the diet, and it's negatively related to body size. We have the log of body size on this axis, and we have a really tight line here. Impressive. This is the percent essentially of leaves in the diet. Small-bodied animals, 90% leaf. Big-bodied animals, 40% leaf. Okay, so this is data to show you that the Jarman-Bell principle is not something I just pulled out of my back pocket. All right, we need to explore the field of allometry, allometric scaling, to understand why this pattern is found in grazers. So the pattern was observed in the field. Now let's link it to some physiology and chemistry that we've been already discussing. So the field of allometry, it's the study of how an attribute changes with body size. I just gave you one. This is an allometric relationship. It's an attribute scaled to body size. Okay? So we're going to help you realize that not all things change with body size. One allometric or non-allometric relationship is something we call scale independence. That's an attribute that does not change with your body size. Here are some examples. For example, the blood cell size. Elephants don't have bigger blood cells than shrews or you and I, which is a fascinating idea. Why not? Why did evolution not select for blood sizes to be different? That is called something that's scale independent. For our purposes today, we want you to realize that the bacteria in the hindgut of an elephant and the bacteria in the foregut of a zebra, I'm sorry, of a cow or an antelope, zebras are hindgut, are the same size, and they probably ferment at the same rate. So that's a scale invariant thing that does not change, scale independent. A trait that we call isometric, you might remember iso means the same, isosceles triangles have equal sides. This is proportional to body size. So for example, in a second, I'll show you a slide that shows your gut size is in general proportional to your body size. Okay, so elephants have bigger guts, but the same proportion to their body size as, say, a small antelope. Leg length is often proportional to body size. So we call these isometric. Do they change with body size? Yes, but they change in a very predictable, proportional way. That's compared to or contrasted with what we mean by an allometric relationship. This is an attribute that changes with the body size due to some kind of mathematical rule. It's not a one-to-one relationship, okay? And I'm going to give you a few examples. The two that we'll focus on today is metabolic rate and the percent of high-quality food in the diet. Let's deal with metabolic rate first. Please remember that I'm defining total metabolic rate as the number of calories, in this case kilocalories, burned by an animal per hour. Okay? How does that scale to body size? So in other words, how much food does an animal burn per hour in all of its cells combined together? That's what we mean by total metabolic rate. How much food you burn in all of your cells doing cellular respiration, adding oxygen to burn glucose to make ATP, as you might remember from biology. Well, here's the empirical data. By empirical, I mean we went out into the world and we measured this for a whole bunch of animals. This isn't a theoretical assumption. This is one we measured. So let's look at the axes. Here's body size on this axis. Okay, and this is kilograms. So some very tiny animals and some very big animals. Here's a 10-kilogram animal. Here's a 1,000-kilogram animal. So we have elephants way up here, right? Mice and rodents down here. Cold-blooded animals, and these are unicellular organisms, right? Single-celled paramecium and euglena and other crazy creatures. Now, notice this is total metabolic rate, just like I said up here, kilocalories per hour. So look at all these dots. The first thing that should impress you is there's a fairly tight pattern around body size. As you get bigger, obviously, you need more food. Okay, so the line should go like that. That's not surprising. Bigger animals need more food. Okay, not rocket science. But what I want you to do is look at this line. This line has a slope of 1. If the lines, all these dots, were along this line, it would be an isometric relationship. There would be a proportional need for food related to your body size. You would burn about a proportional amount of food related to your body size, be the same as compared to any other animal. But notice all of these lines, by separating them out by warm-blooded and cold-blooded and unicellular, we can even get more kind of precision in the data, so to speak. The slopes of those lines are 0.75. Excuse the train. The slopes of the lines are 0.75. That means that this is an allometric relationship. That means that bigger animals, listen carefully, bigger animals burn more food, but less than you'd expect by their body size, because this line goes beneath 1. If the line were above 1, bigger animals would burn more food per body size, but more than you'd expect by their body size. Another way to state this is big animals are more energetically efficient than small animals. Okay? They burn less food per gram of their own muscle mass. All right? This is called Kleiber's law. Max Kleiber, a UC Davis professor. There's a building on the campus. This is where I got my PhD, as you might recall. He was a famous biologist, and he noted that this pattern exists with all kinds of creatures. It is one of the fundamental laws in biology. Yet I want to impress upon you, we don't know exactly why. Right? He said it is not related to surface area. It is not because big animals have a higher surface area to volume ratio. There are other explanations for why this pattern exists, but for now I just need you to know that Kleiber's law is this empirical relationship where bigger animals burn less food per their own tissue than you would expect by their body size. They are more efficient. So what does this mean for quantity and quality of food that an herbivore might need? So as I begun to suggest, bigger animals need more food than smaller animals. In other words, the shape of the line is positive, but less than you'd predict by their body size. They are metabolically more efficient per gram of their own muscle, of their own tissue. So in the long-term scale of things, if animals are competing for limited energy, evolution should select for increased body size through time. And in many cases it has. Think of the dinosaurs, for example. We had much, much larger body sizes at one point in Earth's history. We'll come back to that maybe at a later time. All other things being equal, a 1,000-kilogram animal needs less food than 1,000 kilogram animals. Okay? These are smaller animals. They are going to burn more food per their body size than you would expect from their body size, right? So 1,000 kilogram animals are going to use more food than one 1,000 kilogram animal. All right, now this begins to help us understand the Jarman-Bell principle. But before we get there, we've talked about how much energy they're burning. That's Kleiber's law, their total metabolic rate. But how about how much energy they're gaining? Like, how good is their digestive tract at digesting that energy so that they can get it to their cells where it's burned through the process of cellular respiration? Well, we could make a chart. We could say, well, on the axis of body, on the x-axis, let's put body weight, and our allometric scaling relationship now will be just the size of the gut, the size of either the hindgut or the rumen. Okay, in this case we have both. All right, we have foregut fermenters, and we have these non-ruminants. For a second, don't worry about the difference between foregut fermenters and ruminants, but here are our hindgut and here are our foregut. All right, so they're all on this line together. And notice when we fit a line to all these points, we find that there's an exponent. You might remember from a power law. This is the form of the line. Y equals mx b. B is the exponent. B is the slope. The slope is almost perfectly 1. So this is an isometric relationship. And bigger animals, do they have bigger guts? Yes, but they're proportional to their body size. That's a key point. So on the output side, small-bodied animals are burning more energy proportional to their body size, but they're digesting a proportional amount to their body size relative to big animals. All right? So here's what that means when you put these graphs next to one another. Bigger bodied animals need more food but less than you'd expect, and they have a proportional digestive tract that is really, really, really big, which means the retention time is really, really, really long. So when we put an allometric relationship together with this isometric relationship, this is the food requirements and this is basically the food provision, how much fermentation it can do per unit time, we gain some insight into the Jarman-Bell principle. So what it means is the following. Why do smaller bodied animals need higher quality food like we observed in the field with the Jarman-Bell principle? Well, because they're small, they have small guts, right? Because they have shorter hindguts in some cases if they're a hindgut fermenter, or smaller rumens if they're a foregut fermenter. They can't keep the food in their body very long, maybe a couple hours. Well, what that means is because they have shorter guts, they have shorter fermentation times, they can't digest the really nasty cellulose-rich food. They have to digest the materials that are very soluble, that are really, really high quality. Okay? Now, when you combine that with Kleiber's law, it even becomes a more pronounced selection pressure. Because if you think about going back to Kleiber's law, remember, a small-bodied animal is burning more energy per gram of their own body. And they have a smaller retention time, shorter retention time. So combining those things together constrains these animals to really forage only on the most nutritious, easily digestible food. Okay. So at times you may have to pause and rewind and make sure that you're piecing together the logic here in this presentation, and we will spend some time discussing it when we meet either virtually or in person in NC19 on Monday. Let us return now to our discussion of heterogeneity, which we began on Friday. Heterogeneity of food quantity and quality promotes the coexistence of these animals of different body sizes. That should say sizes there. In other words, each body size has a unique set of requirements for food quality and food quantity. The bigger you are, the more quantity you need. The smaller you are, the more quality you need. Okay? So if we created an environment where the heterogeneity, the change, the variability of that quantity and quality was really high, that might suggest that different animals will always find an opportunity to get the food they need. Okay? But also remember, this heterogeneity is scale dependent to the ruler we're using to measure it. And in our case, ecologically, body size is the ruler. So while small-bodied animals are constrained to eat high-quality food, they're also more able to find high-quality food, which is really rare. Okay? So a couple examples might be that gazelle can find just the living live tissue on the plant, which might be ignored by a zebra or a giraffe, which cannot forage at the same fine scale. So, in conclusion, patterns of body size suggest that evolution has created many body sizes, each of which is a good strategy. There are trade-offs, though, related to an animal's body size. You know, being small might mean you can find high-quality rare food more, but it also means you're being small, you're also constrained to just eat that high-quality food. Being big means you're more energetically efficient, but it also means you need more food, and you've got to move across the landscape and perhaps migrate. So maybe small-bodied animals have smaller home ranges to find rare food in a localized area, but big-bodied animals have to move across the landscape to follow the rain where the high quantity is available for them. We'll learn in more lectures in the coming days that variation in this body size in a way then reduces competition between these species. Okay, they're not all competing for the same resources. It looks like they're all living and coexisting on exactly the same resources of grass and trees, but they're actually sharing the resources because some of the species are best adapted to eating the plants in a particular form. And so because we have rainfall variability in time and space, we have seasonal changes in rain, we have fires moving across the landscape, we have herbivores in themselves influencing the quantity and quality, all of this creates a lot of heterogeneity through time and space. So there's always a time period, there's always a space where these animals can find the kind of food that is required for their physiology. And this is a reminder that what happens at the cellular level actually creates these ecological patterns. If bacterial fermentation were not isometrically scaled to an animal's body size, if the rate of fermentation, in other words, if bacteria that lived in elephants broke down material differently than bacteria that lived in gazelle, we might not notice this pattern. If gut size were not isometric, we might not notice this pattern. if Kleiber's law weren't true, we might not notice this pattern. So these ecological patterns we see are actually what we call emergent properties that arise out of the complexity of the cellular scales. So, in conclusion, when we think about the heterogeneity of a landscape, we have to define it in terms of what we call functional heterogeneity. Function suggests that that heterogeneity serves a role, serves a purpose. So how does the heterogeneity serve the function of being food for a particular animal? Well, it varies. So the variability in a plant in terms of its quantity has to be defined for a particular animal because it perceives that quantity at different scales. And so we can't just, in ecology, just look at the landscape and define it as being heterogeneous or homogeneous. We need to say, well, what's its functional heterogeneity? That means at a scale of an elephant, what does it look like? At the scale of an ant, what does it look like? This opens up the way we do ecology and perhaps even the way we view the world as humans. Now, the rest of this PowerPoint presentation is available for you on Moodle, and I have added a bunch of slides here that I think are interesting. They're not required, and you're welcome to go through them and see if they help you understand some of the themes I've been talking about. But for the video portion, I'm going to end it now, and you're welcome to return to this and look at a later time.